5, 6, 2, 3. This one? DNA, chapter 9, DNA based information technologies. It's very related to what we have learned in the last lecture, which is nucleic acids. And after now, I just finished the other part, which is lipids. Okay, so these two are related ones with nucleic acid and then DNA based information technologies. This will include DNA fingerprinting, agarose gel electrophoresis, DNA microarrays, maybe sequencing, and few other techniques based on what you have learned about nucleic acids. Okay, so all the information technologies are advanced method based on DNA or RNA or any nucleic acid. So let's discuss some of the basics of this chapter, and we'll go ahead and learn the application part. Okay, so by end of this chapter you will be able to learn about DNA cloning techniques. Anyone who have heard stories of clone of people, like real people? I don't want to say the names, it would be political, but yeah, like, have you heard of like clones? Maybe some stories, that you can have your clone, and some big leaders have their clone, and clones are running the country, and so on, all the stuffs. Even Trump has a clone and he's running the country, or I don't want to say the name, but yeah, but you have heard those stories, right? That people can be cloned, like there could be a clone person who could do everything like the same person, and he could sit behind and do the everything so that he couldn't get killed also. So of so yeah, these are the yeah, I can do that. Of yes sir. Yes sir, I always okay sir, okay sir, okay sir, I'll take care. Okay sir. Oh my god, horrible. Okay, he said no extra credits. Okay, we will figure out that later on. Okay, so yeah, DNA based information technologies that includes DNA cloning, maybe cloning of entire human being or entire organism. Organism cloning is already available, like Dolly. You remember Dolly ship? The first clone, not double E. So Dolly sheep, and she was cloned for the first time, like an entire clone. It's possible, it's doable. It's not ethical to do so, but it's doable. You can have a clone of your body, but it's not ethical to have a human clone. Okay, and it will take time for sure. You are like 20 or 25 years old, so it will take again 25 years to grow your clone. So that's physically or scientifically possible, but yeah, depending upon if you are interested or if it's ethical or not. There's a new concept that is already in the market. We call it designer babies. Anyone who have heard of this term before? Designer babies. Anyone who know this thing? CRISPR-Cas9, CRISPR technology. CRISPR technology is a gene editing tool. Okay, so what it will do, if you are very good at painting, she is very good at dancing, or some skill sets you have, so I can combine all those skill sets having the particular segment of DNA responsible for that skill. Okay, and then I can combine that, make a whole human genome, who is an excellent dancer, painter, actor, scientist, doctor, and I can put all those genetic information in a zygote before they are conceived. Okay, so that baby will have all the characteristics you wanted. Okay, and you can design your babies. That's why they are called designer babies. Like the way you select your clothes or anything, you can design your baby. You want him or her to be a cricketer or a dancer or a scientist or a doctor. You can put all those genes responsible that has, you know, all those characteristics, and you can design them. So we'll talk about the tools we can use to cut the piece segment of DNA responsible for a particular function, and also related to how we can express them in the form of proteins. Expression of recombinant proteins. Re means again combination, is combining. So if you combine two different things, we can call that as a recombinant. If it's a protein, recombinant protein. If it's a DNA, recombinant DNA. DNA analysis methods, we'll learn more about that, and DNA microarray. So today I'll try to finish uh, DNA cloning techniques. Okay, and in the next lecture I'll try more focus on the application part. Okay, so it would be interesting to have both the lectures, and it will definitely provide you insight of how we can use modern day techniques to edit DNA or gene or anything else. Okay, so what is a recombinant DNA? As I said, re means again combination, means making a new combination. Okay, and DNA is of course deoxyribonucleic acid. Artificial DNA can be created when you combine sequences that do not occur together in the nature. Okay, suppose if you want a fish to fly, which is not in the nature, okay, you need genes responsible to grow wings, which is in the birds, right? And you of course need gene for the fishes. So if you combine those two genes, maybe your fish could fly. Okay, which is not again making a sense, but anyway, you can combine those two genes together and make a new individual with those recombinant DNA. Okay, forget about the flying fish, move on to the real life examples. Suppose you are diabetic, okay, or you are having a Down syndrome, okay, or maybe someone has a Down syndrome. So we can combine a segment of DNA from a healthy baby to the entire genome, which is having a deficit of the healthy gene from the normal individual. Okay, and you can combine those two genes to make it a complete DNA without any disease. Okay, so it's feasible if you use recombinant DNA technology to cure diseases or to make something which is in need using the molecular biology techniques. Okay, basis of the molecular biology techniques: molecular cloning of genes, overexpression of proteins, transgenic food or animals. Okay, of course transgenic food is in the market. There are a lot of transgenic food. Like earlier days, we never used to have like this big watermelon. Okay, if you go to Walmart or Sam's Club, you see this is a huge piece of watermelon. It was not there before. We used to grow small things before. Okay, and now I think he wants to go to Walmart. So yeah, so we combine genes and making transgenic food. Basically, if you have seen corns, they're transgenic, and even rice varieties. Many of the rice varieties are transgenic. Even we are making artificial meat and other stuffs just coming into the market. Vegetarian meat, vegan meat, or a lot of other varieties. All of them are somehow a product of recombinant DNA technology. Okay, of course animals, we do use transgenic animals for the research purposes. So what we use in research is called humanized model. Okay, suppose I want to study, I do work in Parkinson's, so we can humanize a rat model into a human mimicking model, and study Parkinson's disease. Okay, we can say, hey, this is rat, but all the genes he expresses are human genes. So we can do experiments based on treating rat as a human being. Okay, so here rat identifies as human being, and we can do all the experiments based on that. Okay, so transgenic animals and food, it's there. Gene cloning, as I said already, organism clone. You can clone a whole organism like Dolly or any other, and you can create a genetically identical copies of the organism. Okay, in plants it's very easy. In plant, creating a genetically identical copy is much more easier than animals due to less complexity. The organism may not appear identical, maybe due to regulation of gene expression during development, but you can almost create an identical copy of the organism. Okay, gene cloning is again having an identical piece of DNA. You can isolate from the source organism and amplify to the target organism. As I said, you need a flying fish. Okay, so you will get a gene of interest or DNA from a bird. Okay, and you'll try to incorporate that into a fish, and then your fish might have wings. I don't know how to draw wings, but maybe. Okay, so basic steps: you have to cut the DNA from the bird, and you have to carry this DNA in a vehicle. I call it vehicle, and then vehicle or vector. Then insert the recombinant DNA into the host. You can insert that into a fish, and let you can produce multiple copy of recombinant DNA. Okay, it's doable. Let's understand about what is DNA cloning. Step 1, to generate recombinant vector. Okay, so why we are so interested in making this recombinant vector? Okay, it's very interesting. Okay, as I said, you can make identical copies of yourself, but the problem is lifespan. Okay, our lifespan is almost 80 to 100 years, so you cannot wait for 80 to 100 years to finish your experiments, right? That's why you need something who replicates very fast, mostly the organism which has a life cycle of 20 minutes maybe, like Escherichia coli, which is a bacteria. Okay, so if you want something to see whether it's working or not, you can do an experiment using those animals which has a shorter lifespan. Okay, then you can get the results within 20 minutes rather than waiting for 80 years. Okay, so this is why we are more interested in creating a recombinant vector to understand the fate of any gene or function of any gene, and this is how we reduce all other applications of recombinant DNA biotechnology. So we need a eukaryotic chromosome. Suppose this eukaryotic chromosome is made up of DNA, right? Chromosome constitute DNA, and this piece of DNA has something which is called gene of interest. Okay, gene of interest is what you want. So imagine if you want a gene for insulin. Okay, so you will take insulin gene from the whole eukaryotic chromosome, which is indicated here in red color, and you'll try to see if you can ligate that in a vector. Vector is like a vehicle. Okay, like if you are at your home and you have to come to school. Okay, and this is you. How can you come to school? There are different ways. You can drive a bicycle, you can come by car, you can come through UTEP transport, or any other thing. Okay, all the other uh, transport vehicles you are using will act as vector. Okay, vector is vehicle for the DNA to travel. Okay, and you are the DNA or you are the gene of interest. I am interested to harvest from the organism or donor. Okay, so DNA fragment of interest is obtained by cleaving chromosome with a restriction endonuclease. I introduced a new term which is RE, which is restriction endonuclease. Okay, this is a very interesting name. endonucleases. It's a group of enzymes, and they are called endonucleases. There could be two types of nucleases. I Let's say endonucleases, okay, and the other group which is exonucleases, okay. So suppose I have a segment of DNA. This is a segment of DNA, okay, and there are two enzymes, exonucleases and endonucleases. The difference of their function is, if this is a exonuclease, it will cut the piece of DNA in either sides, okay. If this is a exonuclease, they will cut it from here or here. They are exonucleases. I already told you once, if you are aware, ases means enzymes, like DNases are DNA digesting enzymes. RNases means RNA digesting enzymes. So if I say nucleases means, speak up. If I say nucleases means nucleic acid digesting enzymes, okay. If I say proteases means protein digesting enzymes. If I say lipases means lipid digesting enzymes, okay. So based on how you name them, you can already know the function. Nucleases means nucleic acid digesting enzymes, okay. Exonucleases means they cut DNA sequence at ends, okay. If I say exonucleases could be 5 prime or 3 prime, do you understand what I mean? If I say this is a DNA double helical or single, if I say this is double helical DNA structure, and they would have a 5 prime and 3 prime end, or maybe 5 prime to 3 prime anti-parallel. If an enzyme which cuts the DNA sequences somewhere here, what would you call them? To be more specific, to be more specific, 5 prime exonucleases, okay. So this is 5 prime exonucleases, and if I cut somewhere here, 5 prime, no, yeah, yes, 5 prime exonucleases. If I cut somewhere here, 3 prime exonucleases, okay. This one 3 prime exo. Now suppose if an enzyme which cuts the DNA may be somewhere in the middle, what do you call them? Endonucleases, okay. So up to now you know the differences between exonucleases, 5 prime exonucleases, 3 prime exonucleases, and endonucleases. You do not know the polarity of endonucleases because it could be anything, okay. There is no specific polarity in that. But anyway, let's understand one more term which is called restriction endonucleases. What are restriction endonucleases? It's a very great question you might face. Restriction endonucleases, okay. So let's go to bacteria and see how they work, okay. If you go to a bacteria, they have mesosomes. They do not have a nucleus because they are prokaryote, and this is their DNA, okay. Suppose this is their DNA which is already floating around. You don't find a nucleus which is you do not find a confined nucleus, but this is their, they have one single chromosome which is, okay, and this is their DNA, okay. So bacteria has a different mechanism to protect themselves, okay. We are afraid of bacteria, but bacteria are also afraid of some other creatures, okay. You might have seen them in some scientific movies or maybe some videos. Bacteriophages looks very fancy, okay. They look like this. Anyone who have seen this before? What are they? Kind of. They are called bacteriophages. Okay, bacteriophages are viruses. Yes, they are bacteria attacking viruses. Bacteriophages. Phage means eating. Okay, bacteria eating viruses are called bacteriophages. So bacteria, what did I say? Eating viruses or whatever. Attacking bacteria attacking viruses are called bacteriophages. Okay, so they insert their DNA into the bacterial host and they will try to incorporate into the bacterial host so that they can hijack their system and use them for their own replication to grow more and more bacteriophages. Okay, they want to make babies by using the machinery of the bacteria. Okay, but bacteria are very smart. That's why they are inside us and they are doing all the chaos or disease or whatever, right? So they have a defense mechanism to protect their DNA, which is called DNA methylation. Okay, so they have added methyl group to their whole DNA, which is CH3, CH3, CH3. Okay, and I call this methylation. This is not written in the slides, or this is just the general concept to understand what are restriction endonucleases. Okay, so they have protected their DNA by doing the methylation. Okay, which is which is addition of methyl groups to the DNA, and those methyl groups protect them from the bacteriophages. Okay, so these enzymes are present, restriction endonucleases are present in bacteria. Okay, why they are present in bacteria? As a defense or protective mechanism against bacteriophages. Okay, and they are called molecular seizures. They are called molecular seizures. Molecular seizures means they'll cut the DNA into several pieces, okay, in a specific sequence, and they restrict the growth of bacteriophages. That's why they are called as restriction endonucleases. Why endo? Because they chop their DNA somewhere in the middle. Okay, that's why they are not exonucleases but endonucleases. They restrict the growth of bacteriophages. That's why they are called as restriction endonucleases. Okay, but for the first time, we never knew we can use this machinery of bacteria for our benefit. Okay, how? Because those restriction endonuclease cuts the DNA at specific sequence. Okay, so when you are going to do that, it will take a lot of time, and maybe you will not be able to do that. Why? You have to go to DNA level and you have to cut the phosphodiester bond, which is not very easy to do. So can you imagine how much time it took for us to understand the DNA double helical model or even structure of DNA? Do you think it's possible that you can cut the phosphodiester bond very precisely? It's not possible, but they can do that. Why? They have a machinery to protect themselves against the bacteriophages, and that's how they developed restriction endonucleases for their own benefit. But we humans exploit any other thing which is present in the planet Earth, and we exploited this machinery of bacteria having restriction endonucleases. Okay, we harvested and purified those restriction enzyme, and we used those restriction endonucleases for our experiments. How? Suppose I'll take an example of restriction endonucleases, which is EcoR1. Okay, is this yellow color visible? Let's change it to something else so that look more bright. Green. So let's take example of restriction endonucleases, which is EcoR1. Okay, echo R1 cuts the DNA at a specific sequence called GAATTC. Okay, GAATTC. Okay, this is the sequence of DNA. This is 5 prime to 3 prime, and this is 5 prime to 3 prime. Arrowheads are like this. So can I call the sequence as palindromic sequence? Anyone? I have discussed what is a palindrome. Palindromic sequence. Like you said, words are palindrome, like dad, like mom, or I heard a couple of more words that day. So these are palindromic words, and these are palindromic DNA sequences, which means if you read from 5 prime to 3 prime and 5 prime to 3 prime, in both the directions it looks the same. Okay, so there are specific regions where echo R1 cuts the DNA. Okay, and those specific regions are G and A. Okay, so echo R1 cuts the DNA at a specific position, which is between guanine and adenine. Okay, so imagine if they cut the DNA sequences here, what will be your final sequence after they cut the DNA? You will have one strand called G C T T A, like this, and you have another strand A A T T C, and then G. Okay, you see that? So this is after you treat them with echo R1. So they are called restriction endonucleases, not because they cut DNA at a restricted site, but their ability to restrict the growth of bacteria, bacteriophages in bacteria. Okay, this is more conceptual if you understand why they are called so. Why they are called restriction enzymes. Restriction enzymes, I will repeat the sentence again so that it's more clear. Restriction enzymes or restriction endonucleases are called so not because they cut DNA at a restricted site, but they are capable of inhibiting or restricting the growth of bacteriophages in bacteria. It's a defense mechanism for them. Okay, but they do cut DNA at a specific or restricted site. Okay, which is GATTC for echo R1. There could be many other restriction enzyme, PVU1, PVU2, HIND, HEND2, and there are multiple. Like there are number of restriction enzymes available till today. Okay, that cuts DNA at a different specific site. Okay, but they always cut DNA at a specific site. So you have to remember that. So this is more conceptual. Let's move back to the slides, and you will be able to understand from now onwards. Okay. What are restriction endonuclease? They cut phosphodiester bond. One more thing, they cut phosphodiester bond. What is a phosphodiester bond? We discussed that in last class. Between two nucleotides. Between two nucleotides, you have a phosphodiester bond. Suppose this is a nucleotide with a sugar, with a nitrogenous base, and a phosphate group. You will have another sugar, nitrogenous base, and phosphate group. Okay, this phosphate group and this OH will combine and will make phosphodiester bonds. Okay, so where do restriction endonucleases cuts the DNA? At phosphodiester bond. Okay, and what is the endonucleases, what is exonucleases? You already know that. They are common in bacteria, and they eliminates infectious viral DNA. And what is infectious viral DNA? Bacteriophages. Okay, some make sticky ends, some make blunt ends. What do you mean by blunt end and sticky ends? Okay, it's very obvious from the name. Blunt means it's an end. Okay, you will not have anything hanging around that. Okay, and sticky means you have something hanging around. Okay, suppose if we see echo R1. Okay, for example, echo R1 making a blunt end or a sticky end? Is it a blunt end or a sticky end? Why we call it sticky? Because they have hanging sequences. If you give additional nucleotides or if you just leave them alone, they will again recombine to form the sequence of DNA. That's why they are called as sticky ends. Okay, so eco R1 always makes sticky ends and not blunt ends. What is a blunt end? Suppose if a restriction endonuclease cuts the DNA somewhere here, what is the sequence they will make? GAA, CTT, TTC, and AAG, like this. Is it a sticky end or a blunt end? Blunt, because there is no hanging sequence of nucleotide around the DNA. Okay, so this is a blunt end. Okay, so that's what it was asking. Key that some makes staggered cuts and make sticky ends, some make straight cuts and make blunt ends. Okay, so depending upon what you wanted, you will use those restriction enzymes separately. Okay, large number are known, commercially available, well documented. We have a database. If you want, go and check them out. Let's discuss one of the restriction endonucleases. I think they already discussed the one we have said, which is eco R1. Okay, and if you remember, it cuts the DNA sequence at GAATTC. Okay, GAATTC. Okay, let's zoom in and see if they have the same sequences as I was discussing, right? So it has GAATTC. Can you see that? The next opposite strand also has GAATTC. So what do you call this segment of DNA which has repetitive nucleotides? We call this a specific sequence, which is short chain repeats or something different. This sequence is called palindromic sequence. Okay, so you have to identify palindromic sequence first in order to use any restriction endonucleases. Okay, so you have another palindromic sequence here. Can you see that? Which is CAG CTG, CAG CTG. Okay, now we are using two restriction endonucleases. One is eco R1 and one is PVU2. Okay, and both are restriction endonucleases. And the way they cut here, and this one cuts over GAATTC. Okay, which is this one and this one. So my question is, which of the restriction enzyme makes a blunt end and which one makes a sticky end? Eco R1 always makes sticky ends and PVU2 makes blunt ends because there is no additional nucleotides hanging around. Okay, so I'm clear up to here. So imagine if you have a piece of DNA from here to here and you want this DNA because this is something important. Okay, imagine this DNA is for intelligent quotient, very important DNA. Okay, and you want your child to be very intelligent like Elon Musk so that he can make a lot of money, send people to Mars or whatever. Okay, so you want that gene, you are very interested in that gene. That's why we call that as gene of interest. Okay, gene of interest. So to get that gene, you have to use two different restriction enzymes. One is eco R1 and other is PVU2. Okay, so how can you carry that gene? Is you will use a vector. Okay, vector is responsible for transport. Vector is responsible for transport. Vector is a kind of vehicle that loads the DNA and transport it to the host organism. Okay, so imagine if you want to transport food from this building to biological sciences, you will carry a cart. Okay, and that cart is vector here, which carries DNA. Okay, let's move ahead. We'll use an enzyme which is called DNA ligase. Okay, we have a word in English called ligating, which is attaching. This ligase is an enzyme for DNA. That's why they called as DNA ligase. Okay, what they do, they will have this cut section of DNA and they will add that here. Okay, that's the function of DNA ligase. So if I ask you a simple question, how many enzymes and what are those enzymes required to make this form of DNA, and we can call this DNA as recombinant DNA. Okay, this is a vector and this vector is mostly plasmids. Plasmids are extra nuclear double stranded circular DNA. Okay, that's the definition of plasmid. So I said bacteria, they have mesosomes and they have nuclear material, but apart from that they have a very interesting DNA molecule out, not extra from the nucleic acid or nucleic or uh, or DNA they have. This is extra chromosomal. I already said that extra chromosomal double stranded circular DNA. Okay, extra chromosomal double stranded circular DNA are called plasmids. Okay, and these plasmids are very important. Why? Because you know that we use a lot of antibiotics to kill bacteria. You know that, right? We use antibiotics to kill bacteria. So bacteria has antibiotic resistance genes in those plasmids. Okay, and that's how they evolve and they are not killed by the antibiotics. So this extra chromosomal DNA has antibiotic resistant genes. Why we are telling all this story? Because it will definitely make a sense when we will go ahead with the lecture. Okay, so this vector has now the gene of interest. This is GOI, which is gene of interest, and we have a recombinant DNA available for the next steps. Okay, so imagine if you want to have a recombinant DNA with a gene for insulin, you can cut those sequences using restriction enzymes from the target organism, and where there is your gene of interest, and you can combine them into a vector which is a plasmid. Okay, and now you have recombinant DNA available in the form of vector plus gene of interest. Okay, and this is called recombinant DNA. Let's move ahead and see what's the different types of ligases. Ligases are present in human DNA and bacterial DNA. The only difference is they use ATP and bacteria we use ATP and bacteria use NAD. Okay, anyone who is familiar with NAD? Niacinamide adenine dinucleotide. Okay, if not, it's niacinamide adenine dinucleotide. Okay, anyway, this is another form of nucleic acid, another form of nucleotide. Okay, niacinamide is vitamin B3, I think. Okay, but anyway, this is where vitamins are required. Okay, maybe your DNA ligase of bacteria. Anyway, the process of DNA cloning, which is second step to of the recombinant DNA technology, DNA cloning. We already made DNA recombinant vector. You remember? We made a recombinant vector and we used two different enzymes. One is restriction enzymes, restriction endonucleases plus ligases, plus we used a plasmid DNA and we used gene of interest. So this recombinant vector we use to make this recombinant vector, we use restriction endonucleases, we use ligases, we used a vector as plasmid, and we use the gene of interest what we need to express or need for the recombinant DNA technology. So they have so suppose your host is a E. coli. Host is an organism which is finally responsible for expression of your DNA. Okay, what do you mean by expression? Is suppose if you have a DNA, okay, your DNA will make a copy of mRNA like this, and your mRNA will make protein, and this is central dogma of molecular biology. Your DNA will make RNA, or RNA will make protein. So if you have recombinant DNA, recombinant DNA will express and will uh, produce more and more mRNA, and ultimately more and more protein, and this is what you need. So all the steps you are going to use a system which is responsible for expressing those proteins, and we call them as host. Okay, and we'll use E. coli as host. Why E. coli? Why not humans? Why do not take just all the recombinant DNA and put that in human beings? Because E. coli can replicate in 20 minutes. They can make another copy in 20 minutes. Okay, another copy in 20 minutes, another copy in 20 minutes. So if you need insulin, you can have tons of insulin within hours. Okay, if you keep them in day. So what, how do you think they are making protein in labs? They are using bacterial host to express more and more protein. Okay, make sense? Why not using big animals? Because of their lifespan. Okay, antibiotic selections. You look more serious than me. Is it okay, or I'm going too fast? Are you not understanding anything? Can interrupt me in between. Okay, antibiotic selection. Where are antibiotic genes are present in bacteria? I said bacteria has antibiotic resistant genes. Okay, there's a lot of stuffs going on in antibiotic research. Okay, why? Because bacteria are smart. They're smart guys for sure. They are more smarter than us. That's why they have molecular seizures, restriction enzymes. They methylate their DNA, and they do every other thing possible to protect themselves. So if you are treating them with antibiotic every single day, okay, if you have a fever, you just take antibiotics. What do you think? They are not evolving? Of course they do. That's why where it comes antibiotic resistance. Okay, so now most of the bacteria are antibiotic resistance around us. Okay, so we need more and more new antibiotics in years to come to protect us from those bacteria. They are not going to be harm. Those antibiotics are not going to kill bacteria anymore, because they are resistant now. Most of the antibiotics we are using now, most of the bacteria are now getting resistance of those antibiotics, and those genes are present in their plasmids. Okay, there's extra chromosomal circular double stranded DNA is plasmid, and those bacteria are getting resistance to the new antibiotics we are making. Okay, so that's where it comes the role of antibiotics. Okay, so as discussed, antibiotics could be different. If you heard, this is the most famous antibiotic, which is penicillin, which is discovered by accident, serendipity. So penicillin, ampicillin, chloramphenicol, these are the few antibiotics available in the market, and now bacteria are getting resistance to those antibiotics, and those are not helpful anymore. Okay, so we need to develop more and more antibiotics now in order to protect us from those bacteria. Okay, so plasmid can carry genes that gives host bacterium resistance against the antibiotic, and allow the growth of bacteria that have taken the plasmids. Okay, so imagine I'm taking a plasmid. Okay, this is a plasmid, and this doesn't have any gene for antibiotic resistance. Okay, this is just normal plasmid, but I have antibiotic resistance gene somewhere. Okay, example for penicillin, AMPR means ampicillin resistance. Okay, and this is a piece of DNA which has ampicillin resistance. Okay, I will use recombinant DNA technology and you will use restriction endonucleases to cut this piece of DNA from the organism that has antibiotic resistance, and I'll add this using DNA ligase. Okay, I'll cut this also with the restriction endonucleases, and I'll add this piece over here. Okay, now you have a recombinant DNA, but your recombinant DNA is ampicillin resistance. Okay, now let's put this in a bacteria. Let's put your antibiotic resistant plasmid in bacteria. Okay, now your bacteria has antibiotic resistant gene somewhere here, and now if you treat this bacteria with ampicillin, do you think they will die? Yes or no? Exactly. Will they die? No. Why? Because they are resistant towards the ampicillin. Okay, so the way we grow bacteria in our lab is we use agar plates. Okay, these are plates like this. If you grow this bacteria over here, they will be able to make the colonies. Why? Even if the medium has ampicillin. Why? Because they are now resistant to the particular antibiotics. Okay, similarly you can add multiple antibiotic resistant genes to a plasmid to make sure they can survive in a condition or environmental condition if they are exposed to different antibiotics. Okay, this is how we can select the colonies which has antibiotic genes. Okay, so let's understand how we can exploit that in selection of recombinant and non-recombinant colonies. Okay, so there are two different terms called transformation, and we'll have transformants and non-transformants. Okay, what do you mean by transformants and non-transformants? Okay, suppose, why would you carry something which is not yours? Okay, I'll not carry your bags to the biological sciences if it's not mine, right? Why would bacteria carry something which is a plasmid DNA which is not theirs? They will say, no, why would we take that? We will just kick them out, and they do that most of the time. It's very hard to incorporate something which is foreign to bacterial uh, to the bacterial cellular bacterial cells, right? So bacterial cells do not take up foreign objects, DNA, RNA, or anything. If they find this is foreign, they will chop them using restriction enzymes and they will digest them, because this is foreign to them. This could be dangerous. So we make fool of bacteria so that they cannot recognize what is a foreign object. Okay, and we try to force them to take up the plasmid DNA. Okay, and we do that using different methods. Sometimes we use gold gunshots, sometimes we heat cold them. So we provide them a heat and then cold them. We call that heat shock treatment. Okay, heat shock treatment, and we heat them for some temperature and we cool them again, heat cold, and then they somehow take up the DNA. Okay, because when you heat them and when you provide cold shock, their membranes, they dilate and constrict, and during that you have pores where you DNA can go in and sit inside the bacteria. And we use different techniques. We do electroporation. Electroporation is a technique where you can literally insert a insert a microneedle to the bacterial plasma membrane and transform them and transfer the DNA. Okay, and there are different methods. Some people use gunshots, the small bullet shots where tip is coated with the plasmid DNA, and you shot them, and they will go inside the bacteria and will do their job. Okay, so these are the forceful methods we use towards bacteria so that they can take up our gene of interest having that DNA. Okay, so anyway, so this is a foreign DNA and this is a vector, as you see, PBR 322, which is one of the very famous vectors in history. PBR. P stands for plasmid. Yes, yes. BR. Anyone remember the name of those two scientists who created this vector? If not, it's Bolivar and Rodriguez. Okay, Bolivar and Rodriguez are two scientists who created this vector, which is called PBR 322, which is one of the most famous in recombinant biotechnology. Okay, so this P is for plasmid, B are for Bolivar and Rodriguez, and 322 is the number of sites you can attach your DNA. Okay, so what's the special about this PBR vector is they have two antibiotic resistant sites of gene. One is ampicillin and other is anyone? Tetracycline. Very good. So one is ampicillin and other is tetracycline gene. So these are antibiotic resistant genes that a plasmid already has. Okay. What's more interesting is when you can you see there are two patches, one is green and one is blue. Okay, and look at this, where do you think restriction endonucleases cuts the DNA, at blue side or green side? Green side, right? And green side is responsible for which antibiotic resistance? Ampicillin. Let's focus and understand what is happening. Okay, so suppose if a particular gene which is responsible for a function is cut in the middle, what will happen? Suppose I'm using this Apple pencil to draw. What will happen if you cut this pencil into half? It will stop working. I cannot write anymore. So what will happen if you cut the piece of DNA which has ampicillin resistance gene? It will no more have the antibiotic resistance against ampicillin. What will happen if I cut the DNA sequence somewhere in the middle of blue? It will be no longer resistance to the tetracycline antibiotic. Okay, and what they have done in this case is they have inserted this piece of foreign DNA, which is represented in red color, into a green site which is ampicillin resistance. Okay, and now they made a recombinant DNA over here. Another ghost. Let's understand recombinant biotechnology today. So we have created a recombinant DNA having a foreign DNA, and also we lost the antibiotic resistance towards ampicillin. Okay, am I clear up to here? Okay, now there are two different cases. There could be a transformant colony and there could be a non-transformant colony. Okay, you want this DNA to be taken up by the cells. Okay, this is called process of transformation. Okay, I could explain them more in detail because I do those experiments. Like I do transform the cells, bacterial cells, trying to put new plasmids. Okay, I try with alpha synuclein protein, which is responsible for Parkinson's disease. So I do have the vectors which has genes for alpha synuclein, and I transform them in E. coli BL21 strain of bacteria to see if they have transformant or non-transformant colonies. Okay, so you can see there are two type of cells, bacterial cells here. One has a vector and one doesn't have a vector. But I could also see a different type which has a vector with a foreign DNA. Okay, so this is one type, this is two type, and this is third type. Okay, and you could not see any more types. Okay, what's the difference between this one and this one? it doesn't have a plasmid. It means they did not take up the vector or plasmid. Okay, they said no, not allowed, no entry. Okay, so they did not take the plasmid. Okay, there are two others. One is having the transformants DNA, and which is 3, which has the foreign DNA, and one without foreign DNA. Okay, so what's the difference between 1 and 3? One is resistance to ampicillin and other is no more resistant towards ampicillin. So suppose I make two separate albi agar plates, okay, and I put ampicillin as antibiotic in both of them. Okay, now I am taking and now I'm sticking a plate for both of them. Okay, so whichever has ampicillin resistance gene will be able to survive on those plates. Okay, if they do not have a ampicillin resistance gene, they will die. Okay, and you will see there are no bacterial colonies on your plate. Okay, makes sense? So we are using two different antibiotics here, ampicillin and tetracycline, but they are resistant towards tetracycline. So let's understand what is happening in the next step. Okay, so how do you select transformed cells? These cells are transformed, these are not transformed. Why? Because they do not have that red foreign DNA. Okay, these ones not at all, they do not have even a plasmid. Okay, these are transformed cells, incorporated plasmid, they have incorporated plasmid within them. This is no. So these two are important to us. So how you can select those transformed colonies? Okay, so you have two antibiotic, one is ampicillin, another is tetracycline. Okay, and I said if you add a DNA in ampicillin resistance gene, it will no more be resistance towards ampicillin, but it will still be resistance for tetracycline because their function is not affected by the insertion on some other sites. Okay, this is a very common term which is very informative term used. I don't see it here, but I have learned in my place, which is insertional inactivation, which makes very sense to this case. Insertional inactivation, do you understand what I mean? So insertional inactivation means when you insert a piece of DNA to a functional gene DNA sequence, their function is going to be affected, inactivated. Okay, so when you insert a piece of DNA, the function will be inactivated. That's why we call insertional inactivation. Okay, so if you have agar plates that has tetracycline gene, of course it will have colonies because it's not affected. But if you have only ampicillin, the colonies will die because they do not have ampicillin resistant gene now. What will happen if you combine both ampicillin and tetracycline? It's mind tricking, but think of it. What if I put a agar plate with both ampicillin and tetracycline? Do you think there will be colonies still going on? Yes, you are correct. So do you think there will be no colonies at all? But I could see some colonies. So you missed a very important discussion here. Is can you see this one? Exactly. Why? Why so? Are you getting what I'm trying to say? Yes. There would be still some bacteria which is not having that foreign piece of DNA. If they do not have that foreign piece of DNA, that means their ampicillin gene is still intact and they are ampicillin resistant. That's why you can see a few colonies because of the bacteria which did not take up the foreign DNA. Okay, that's why you see a few colonies even if you have combined ampicillin and tetracycline. Okay, so I think we are clear up to here. It's a very important discussion. Now let's move to the cloning vectors. Okay, any questions till now? Preguntas. Good question, actually. I would say why? Because when doctor prescribes you antibiotic, they change the course of antibiotic within few months. This is for a few months now. I'll give you a different course of antibiotics. Why? Because if I keep exposing you with the same antibiotic, your bacteria will learn how to cope up with that. They will develop that as their defense mechanism, and all the bacteria in your microbiome is resistant to that particular antibiotic. So suppose if you are coming to this classroom again and again every single day, you know your steps. Even if you are like talking to your phone or doing something else, you will come at the same place and sit over here, because your brains know. Bacteria will also know this is the antibiotic which is coming to me every day. Then I will prepare myself and develop a mechanism against that. But if they come and see a different molecule every single day, they're confused. So imagine they are prepared for the first one and they see a different molecule, and they will say, oh, we don't have to prepare for this because this is no longer available. So let's prepare for this. When they start preparing for this, you will change the course of antibiotic. So there will not be antibiotic resistance if you are not taking a medicine for a longer time. I would personally, I do not prefer to take medicine or antibiotic if I am sick. Even I would prefer 2 or 3 days rest rather than taking antibiotic and getting up the next day. Okay, because let your body fight, let your immune system work for you. Otherwise you are making your immune system not working for you, which is very harmful for long durations. Okay, if you are sick, go to bed, take rest, take leave for 2 days, and your body will fight for that. And that's more useful, because now if they see a new molecule, they will start fighting without even asking you or without waiting for the antibiotics you take. Okay, so it's better to go things with a natural pace. Okay, your body is capable of fighting cancer. Forget about the small viral fevers and bacteria which is infecting you every day. Okay, go for the places and look at the people where they are living in poverty and they are still not affecting by diseases because their immune system is more developed now. So we felt that importance during COVID. Okay, if you are in a good place, you are healthy, living a good lifestyle, not exposed to the allergens or antigens in your early stage of life, you are biologically weak, because your body doesn't know what is a huge variety of microorganisms out there in the world. Okay, so when you are exposed today with the COVID virus, your body knew that virus for the first time. But if you are in a place where you are exposed to multiple pathogens when you are like a child, your body knows that. They have a memory T cells in your body which will be immediately activated when they see the same virus even after 20 years. So it's very important to build an immune system. Let your child play in woods, let your child play in water or mud or whatever, so that they can gain their natural immunity through biological sense. This is how we should be growing up. Anyway, let's move back to the cloning vectors and see what is plasmid. I say plasmid are circular DNA which is extra chromosomal in bacteria. They can replicate autonomously, which basically means they do not need a vector or someone else's order to replicate and make their own copies. So they can replicate autonomously. The next thing is they can carry antibiotic resistant genes, allowing cloning of DNA up to 15,000 base pairs. Okay, so you can only clone a short sequence of DNA using plasmid. But imagine if you want to clone huge DNA, like 300,000 base pairs. You want to clone the whole chromosome, which is a huge structure of DNA, right? You need some bigger systems, okay? If you want to make, if you want to open a brewery, you need bigger tanks to fill in, you know? If you want to make cocktail here, it's easy. You can mix up things, 2 or 3 bottles, that's it. But if you need bigger systems, you of course need bigger machinery, okay? And there comes, we made artificial chromosomes, okay? And now I'm introducing two different terms here, which is bacterial artificial chromosome and yeast artificial chromosome, okay? These are the two artificial chromosomes humans made in order to replicate whole chromosomes or to make clones of larger base pairs, okay? Bacterial artificial chromosome and yeast artificial chromosome, okay? What are they? See, this is a plasmid. This is how a plasmid should look like. Now I think you are able to understand what do you mean by SAL2, BAM-H1, ECO-R1, PST. What are they? They are uh, 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 they are restriction endonucleases. Okay, and what is this AMPR and TET-R? Ampicillin resistance gene and tetracycline resistance gene. And what do you mean by PBR322? P means plasmid, BR is name of scientist Bolivar and Rodriguez, and 322 is they have 322 cloning sites. Okay, what will happen if I introduce a piece of DNA here? What will happen if I introduce a piece of foreign DNA here? Yes, please. Exactly. They will lose their capacity to defend towards ampicillin. What will happen if I add a piece of DNA here? Exactly. What if I do like add foreign piece of DNA in both the antibiotic sites? Exactly. There will be no more resistance toward ampicillin and tetracycline. Okay. What will happen if I add a piece of DNA here? Nothing will happen. Why? It's not affecting any of the antibiotics sites. Okay. Got that? What will happen if I introduce a piece of DNA at ORI? ORI is origin of replication. Maybe it will not be able to replicate anymore, because this is the place where origin of replication starts. Okay, PVU2 is another site. These are all the restriction enzyme sites in the plasmid. Okay, am I clear up to here? Let's understand about BAC and YAC, bacterial artificial chromosome and yeast artificial chromosome. Okay, so I'll try to move fast from now because we understood most of the things here. This is resistant towards CAM. CAM, if you don't know, is chloramphenicol, which is another set of antibiotics. Okay, this has a ORI. This is BAC, standing for bacterial artificial chromosome. And imagine if you have a cloning site which is having a LAC-Z gene. Okay, LAC-Z. Okay, this enzyme's code for beta galactosidase. Okay, this is more recombinant biotechnology stuffs. Okay, but anyway, I'll show you what do I mean by LAC-Z. Okay, so you cut the piece of DNA using restriction endonuclease, okay, and you make a spot here so that you can insert a foreign DNA. You see that this bacterial artificial chromosome is treated with restriction endonucleases, and they will create a large DNA fragment with appropriate sticky ends. Why? Because you are using a restriction enzyme which creates a sticky end. Okay, now you will use the same restriction enzymes to cut a segment of DNA from your gene of interest, and you will add this together with DNA ligase. Okay, and now you have a recombinant bacterial artificial chromosome with this foreign DNA and this bacterial artificial chromosome. Okay, and now you made a recombinant bacterial artificial chromosome because it has a foreign DNA plus bacterial artificial chromosome. Okay, when you insert a gene in ampicillin resistance, the ampicillin resistant property got lost. Similarly, when you add a gene here in cloning site of leg Z, this gene will be disrupted, right? And they will no more code for beta galactosidase enzyme. Why we are more interested in knowing that? Because we should know whether the colonies are transformed or not. Okay, we will do the electroporation. This is a forced method of for bacteria to take up the artificial chromosome now, and by electroporation we inserted this recombinant bacterial artificial chromosome. Okay, now you have a agar containing chloramphenicol and X-gal. Okay, what is a chloramphenicol? So have you seen this chloramphenicol here resistance gene? So it's not affected in this case, so it will still have resistance toward chloramphenicol. Okay, so what is the major factor which will be decided for this is the place where gene is inserted, which is like C. Okay, there's a term called X-gal. Okay, and we have beta galactosidase enzyme. So X-gal is reporter gene, which if they are present, they will make blue colonies. Okay, and if they are absent, they will not be able to make any colonies which is colored, and you will find white colonies. Let's understand, it's very confusing. So if you get a color, your experiment didn't work. If you did not get a color, your experiment worked, which is very unusual. Okay, if we see a color, we are happy. If you do titration, if you see pink color, we are happy, right? But if you see a blue color here, it's not working, because if you have a blue color, it means beta galactosidase is doing their job. If they are doing their job, it means the gene we are trying to insert at that location, it was not inserted. Similarly as penicillin. Okay, if your bacteria is still resistant to penicillin, it means foreign DNA was not incorporated. Okay, similarly if you see a blue colony, means your experiment failed. So if you are seeing a blue colony next day, not happy. If you see a white colony, means your experiment did work and your transformation was successful. Okay, am I clear to here? So X-gal is a reporter gene for forming color colonies. If you see a color colony, you should be sad, and if you see a white colony, you would be happy. If you are more interested, go and read through Lehninger and you will find more information. Let's move on to the yeast artificial chromosome. Okay, don't afraid by seeing this. Okay, very simple, but you have to read through literature. What happens when I run through the slides is I can give you a very little information about the experiments or whatever. I am a big fan of reading textbooks. If you want to have real knowledge, go and read textbooks. Okay, go and read Lehninger. You will find more concepts, you will learn things more deeply, and you will be able to have that knowledge to be written on your paper. Not just because your exams, you will have those conceptual things in you, okay, that you can apply further on. So read books. If you see this, this is just a picture from the Lehninger book, but I can explain you all the way. But if you read book, you will definitely get more in-depth knowledge of the concept. Okay, so this has, now up to here you know what is EcoR1, what is BAM-H1 and BAM-H1. This is again restriction endonucleases. YAC stands for yeast artificial chromosome. ORI is origin of replication. C is another site. Selectable marker Y could be any antibiotic resistance gene. Selectable marker X could be any other antibiotic resistance gene. Antibiotic resistance genes are selectable markers. I'm repeating this sentence again. Antibiotic genes are selectable markers. Why? It helps us to select between transformant and non-transformant colonies. Okay, that's why they are called as selectable markers. Okay, so let's move ahead and see what will happen if we treat them with the BAM H1. BAM H1 will cut this artificial chromosome at two different sites, and it will open up the whole structure like this, and it will create a different segment here, okay, which has telomeric ends. Now you want to use eco R1. We know eco R1 cuts. Eco R1 is another restriction endonuclease which cuts at GAATTC. Now it will create this into two different pieces, and now between these two pieces you can add fragment of foreign DNA, okay, here, and now you can make a yeast artificial chromosome like this, okay. Once your chromosome is ready, you insert them in yeast. Yeast is basically a fungus, so you have to digest the cell wall first. See this cell wall of fungus, and then after digesting cell wall, it is called spheroplast, okay, and then you can transform this piece of DNA into here, and it's available for cloning. Okay, understood? Simple, just follow the steps. And it says yeast are simplest eukaryotes for DNA recombination. They have linear chromosomes with telomeric ends. They can be unstable in vitro for sure, because anything which is floating around is unstable. They are circular to accommodate for quick replication, stable storage, but removable segments can linearize and for product transformation. Let's understand the next thing, which is site directed mutagenesis. Very important, very important. Site directed mutagenesis. I think we have already introduced the term mutagens. You remember, I give a very good example of how to protect your skin cells from skin cancer. You remember, to put sunscreen, and if you do not use your sunscreen, something will happen. And what is that something? That's the physical nature. Let's go to the genetic level and tell me what will happen if you do not use the sunscreen. Dimerization of pyrimidines. You remember that? Can you hear my words from the previous class? If you do not use your sunscreen, your pyrimidines could dimerize. Okay, so that's specific. So imagine if you could use any chemical that could go to your DNA and mutate at some specific position. Okay, that's called site directed mutagenesis. Mutagenesis is process of induction of mutation. Okay, mutagenesis is process of induction of mutation. Site directed means you can literally precisely you can tell them I need mutation particularly in this area or this gene. Okay, so let's go ahead and see. uh, Understanding protein function. So imagine if you have mutated the genes for ampicillin, your property of ampicillin resistance got lost. So imagine if you do not know function of a particular protein, if you do not know what this Apple Pencil can do, just break them and start writing. You will not be able to write it because this function is lost. It is called loss of function. Okay, if you purified a protein and you do not know the property of this protein, go and modify the genes or DNA responsible for production of this protein, and any function in the organism will be affected, and that's how you know this protein is responsible for that function. Okay, if this is a protein responsible for calcium transport, and if you mutated the gene responsible for coding this protein, your calcium transport will be affected, and that's how you know this is particular function of the protein. Okay, so side directed mutagenesis are helpful in providing the function of the protein, and if you change the nucleotide of the coding DNA, the expresses mutated gene has to change. um, they are induced by chemically synthesized mutated primers. Mutated plasmids are always sequenced to confirm that desired mutation is present. Okay, of course you will know if there is a loss of function, that protein is affected. Okay, so not much to explain here. Next thing is PCR, which is polymerase chain reaction. I tried to introduce this in last class. PCR, which is polymerase chain reaction, and polymerase chain reaction is helpful in amplifying the gene of interest. So imagine if you want to detect a disease or a viral infection, and you have a piece of DNA or RNA, you can make multiple copies of the same DNA or RNA, and it will be more easy for detection. Okay, so what do you need? You need target DNA, you need primers, nucleotides, and DNA polymerases. Okay, it's very important to understand DNA polymerases here. They are not normal DNA polymerases. DNA polymerases are enzymes responsible for polymerization of DNA. Okay, so these enzymes are very special enzymes. We again use or exploited bacteria for that. Okay, we cannot have our DNA polymerases functional at higher temperature. That's why people die at higher temperature, because their enzymes are no more functional. But bacteria can do that. So we have thermal springs where bacteria are surviving more than 100 degrees Celsius. They are still there, they are alive, means they have enzymes which are functional at those high temperatures. Okay, and we extracted and purified those enzymes from bacteria which are present in thermal springs or deep sea, because they can stand high pressure, temperature, or anything, adverse environmental conditions mostly, and we use them for polymerization at higher temperature around 95 degrees Celsius. Okay, so let's see, there are three different steps responsible for polymerase chain reaction. The first step is to separate these two strands of DNA. Okay, and this process is called denaturation. The first step of PCR is called denaturation. So if you provide heat, definitely these two strands will separate, and which bond will break? Mm-mm. Which step will break, which bond will break if you denature DNA? Yes, which bonds? Hydrogen bonds. Okay, because two strands are connected with hydrogen bond. Adenine and thymine, two hydrogen bond, and cytosine and guanine, three hydrogen bonds. So denaturation steps, hydrogen bonds are broken if you apply heat to them. Okay, now you will add synthetic DNA oligomers and nucleotide primers. Nucleotide primers are like, if you want to make DNA strands, you have to give the units. It's like Legos, you know. If you want to polymerize, you have to provide those substitutes um, for the nucleotides to form the polymeric chain. Okay, and once you add them, you will add the enzyme which is Taq polymerase. Taq polymerase is important. Why? Because it's a unique enzyme, Taq polymerase. Okay, why? They can work in high temperatures. There's an organism where we go to harvest this, which is called Thermus aquaticus. Thermus aquaticus. Okay, this is name of the bacteria where these enzymes are present. Thermus aquaticus. So this Taq polymerase again will help to polymerize the DNA. See that 5 prime DNA polymerization always happens 5 prime to 3 prime. Okay, so if your strand is initially 3 prime and this is 5 prime, your DNA replication will go in this direction, 5 prime to 3 prime, opposite to this one. And now finally when you have to repeat this step. This step is called, second step is called annealing, where you provide all the materials required for replication. And third step, where you when you are done with the polymerization, it will again, you have to decrease the temperature so that hydrogen bonds can form again. Okay, so you have to repeat this step again and again, and you have one copy of DNA. When you start with one copy of DNA, at the end of the cycle you have 2. If you start with 2, you will have 4. If you start with 4, you have, so this goes in 2 to the power of n. Okay, if you go 2 to the power 8 cycles of PCR, how much DNA will have? 2 2 2 2, which is 4 8 16, 16 multiplied 16 again, how much it will be? 244 or something, whatever it is. Okay, so you will have many number of copies just in 8 cycles of replication. I think next slide is the same thing. If you go to 25 cycles, you have 10 to the power 6 fold of DNA. So imagine, this is a huge number. Okay, that's why I say if you save $1 today and start increasing its multiple of 2, you will have so much money at the end of the month that you cannot imagine. Okay, so this is what we do and amplify our DNA in order to um, detect those DNA somewhere. Okay, if it's viruses or it's a bacteria, any DNA you can replicate and use it for the detection purposes. Okay, the next is separation of DNA strand by electrophoresis, and I think it's time, so we'll stop here and we'll start electrophoresis and DNA fingerprinting tomorrow.